Welcome to the Deep Dive, where we take a stack of sources and extract the most important nuggets of knowledge to get you well-informed, fast. Today, we're embarking on a journey into the incredible world of plants, specifically flowering plants or angiosperms and how they reproduce. And we're actually starting with a story that might sound, well, completely unrelated.
It's the origin story of Velcro. Imagine an engineer, this was back in 1941, hiking through the Swiss Alps. His dog comes bounding back absolutely covered in these little spiky burdock burrs.
Oh, I know those annoying little things.
Exactly. So he pulls them off, probably a bit annoyed, but then his curiosity kicks in. He pops one under a microscope and sees these tiny, tiny hook structures that latch onto the loops of the dog's fur.
Ah, okay.
And that simple observation sparked an idea. After years of development, he created the hook and loop fastener we all know now as Velcro.
I had no idea something as common as Velcro could lead us down such a fascinating rabbit hole. Because what's truly remarkable here is how that seemingly trivial annoyance for a dog reveals a much deeper, more intricate connection between plants and animals.
Mm-hmm.
Those burrs weren't just random hitchhikers. They're designed for one thing, to spread the plant's seeds using animals as, unwitting dispersers.
Yeah.
It's an initial surprising glimpse into the complex world of plant reproduction and, maybe even more surprisingly, how deeply intertwined human ingenuity has become with the plant kingdom.
Absolutely. And today, our deep dive expands to cover angiosperms, the flowering plants. These aren't just pretty faces. They are, without question, the most vital plants on Earth. They form the backbone of agriculture and pretty much all terrestrial ecosystems.
Right.
We're going to unravel their unique reproductive biology, from their incredible flowers and unique fertilization processes to how we as humans have profoundly altered them through domestication, traditional breeding, and, cutting-edge biotechnology.
So our mission today is to pull back the curtain on the ingenious biology of flowering plants. We'll explore what are often called the 3 F's flowers, double fertilization, and fruits, alongside the fascinating world of plant reproductive strategies and how humans have profoundly shaped them over millennia. Get ready to see the plants around you in a whole new light. All right, let's dig into the fundamental life cycle. For anyone who remembers a bit of biology, plants have this unique thing called alternation of generations.
Right.
It's where they essentially live a double life, alternating between a multicellular haploid stage, the gametophyte, and a multicellular diploid stage, the sporophyte.
And for angiosperms, these flowering plants, what's really striking is that the sporophyte is the dominant generation. That's the large, obvious plant you see, the tree, the rose bush, the cornstalk. It's the longer-lived phase.
Okay.
The gametophytes, both male and female, have undergone this remarkable evolutionary reduction. They're tiny, often just a few cells, and they're entirely dependent on the larger sporophyte for all their nutrients. This miniature scale is actually key to their success.
So let's start with the first F flowers. If you picture an idealized flower, it's typically built from 4 types of organs arranged in these concentric rings or whorls.
Mm-hmm.
On the outside, you've got the sepals, usually green, which act like protective little leaves for the budding flower. Inside those are the petals, often vibrant and fragrant, their main job being, to attract pollinators.
The billboards, basically.
Exactly. Next, we find the stamens, the male reproductive organs. Each one has a stalk, the filament, and an anther on top, which is where the pollen is produced.
Right.
And right at the very heart are the carpels, the female reproductive organs. A carpel usually has this swollen base called the ovary, which contains the ovules. Extending up from the ovary is the slender style, topped by a sticky stigma, specifically designed to catch pollen. You mentioned carpels, but I've also heard the term pistil used. What's the distinction there? Is it the same thing?
That's a great question, and it's a common point of confusion. Think of a carpel as a single unit, like a building block. A pistil is simply the collective term for the female part of a flower. It could be just one single carpel, or it could be several carpels that have fused together into one larger structure.
Ah, I see.
So essentially, all pistils are made of carpels, but not all carpels form a pistil on their own, if that makes sense. And this amazing diversity we see in flowers, even incomplete ones lacking parts, like grass flowers without petals.
Right.
It's almost always an adaptation for reproductive success, often linked to a specific pollinator or pollination method like wind.
It's incredible how tailored flowers are for their job.
Mm-hmm.
But how does a plant actually decide to make a flower instead of just more leaves? Is it just random?
Not at all. It's a very precise biological switch. The plant's growing tips, the meristems, get a signal triggered by things like day length or internal plant hormones, and they stop producing leaves and start producing flower parts instead. It's a major developmental shift.
And how does the plant know which part to make where? Like, how does it know to put sepals on the outside and carpels in the middle?
That's where the ABC hypothesis comes in, and it's truly ingenious. Think of it like a genetic recipe, or maybe three different types of paint, A, B, and C. These three classes of genes, the A, B, and C genes, are active in specific overlapping zones or whorls within the developing flower bud.
Okay.
Where A genes are active alone, on the outermost whorl you get sepals. Where A and B overlap in the next whorl, you get petals. If B and C are both active in the third whorl, you get stamens. And where only C is active in the centermost whorl, you get carpels.
Wow.
It's a beautifully precise genetic ballet. And if there's a mutation in just one of these genes, say a C gene isn't working properly, the plant might grow petals where it should have had stamens, or maybe more sepals instead of petals. It's a clear demonstration of how these master genes control the flower's form.
That's a fantastic analogy.
Hmm.
So once the flower structure is set by these ABC genes, how do those tiny gametophytes, the actual reproductive cells, develop inside?
Right, so for the female gametophyte, which we also call the embryo sac, it forms deep within an ovule inside the ovary. A single specialized cell in there, the megaspore site, undergoes meiosis. This produces 4 haploid cells, but typically only one survives.
Only one.
Yeah, the other 3 usually just degenerate. This surviving megaspore then undergoes mitosis a few times, but without cell division initially, creating a single large cell with 8 haploid nuclei. Then, membranes partition this into a 7-celled structure, the embryo sac. This includes the crucial egg cell and 2 other nuclei called polar nuclei in the central cell.
8 nuclei, 7 cells. A bit mind-bending.
It is, but it's incredibly efficient. Everything is perfectly set up for fertilization within this tiny protected structure.
And for the male side, the pollen, how does that develop?
The male gametophytes, the pollen grains, develop in the anthers, those structures on top of the stamens. Inside the anther, specialized diploid cells called microspore sites undergo meiosis, each producing 4 haploid microspores. Each microspore then develops, usually through mitosis, into a 2-celled pollen grain. It has a tough outer wall. One cell is the generative cell, which will later divide to form the 2 sperm cells. The other is the tube cell, which, as the name suggests, is destined to form the pollen tube.
And that journey, of course, is pollination. The transfer of that pollen grain from the anther to the stigma. Once it lands on that sticky stigma, what happens?
If it's compatible pollen, it germinates. The tube cell starts growing, forming a pollen tube, like a tiny tunnel that burrows down through the style towards the ovary. It's actually guided by chemical signals released by the ovule.
Amazing navigation.
It really is. And this tube carries the 2 sperm cells, formed from the generative cell dividing, directly to the opening of the ovule and into the female gametophyte.
And here's where we hit the second F, double fertilization.
Yeah.
You said this is really special for angiosperms.
It is absolutely unique and highly efficient. Once the pollen tube delivers the 2 sperm cells into the embryo sac, one sperm fuses with the egg cell. That creates the diploid zygote, the first cell of the new sporophyte embryo.
Okay, standard fertilization. What about the second sperm?
The other sperm does something equally vital. It fuses with the 2 polar nuclei in that large central cell of the embryo sac. Since the polar nuclei are haploid and the sperm is haploid N, this fusion creates a triploid, 3N nucleus.
Triploid. 3 sets of chromosomes.
Exactly. This triploid cell then rapidly divides and develops into the endosperm. The endosperm is the nutrient-rich, food-storing tissue for the developing embryo. Think of the white, fluffy part of popcorn or the bulk of a wheat grain that's endosperm.
No.
This double hit is incredibly efficient because it synchronizes development. The plant only invests energy in creating the endosperm, that valuable food supply, if the egg has actually been fertilized. No fertilization, no endosperm, no wasted resources.
That makes perfect sense. So after this amazing double fertilization, everything changes. Each fertilized ovule starts developing into a seed,
Right.
and simultaneously, the entire ovary surrounding those developing ovules transforms into what we recognize as a fruit, the third F.
Precisely. Inside the developing seed, that diploid zygote begins dividing, forming the embryo. It develops rudimentary leaves called cotyledons and the beginnings of the shoot and root systems.
And if you look at mature seeds, they show incredible diversity, right? You mentioned different types.
Yeah, absolutely. Take a common garden bean, for example, that's a eudicot. In the mature bean seed, the endosperm has often been absorbed, and the food reserves are stored in the two large, fleshy cotyledons, the two halves of the bean. But now look at a monocot seed, like maize or corn. The food supply largely remains as endosperm. Corn has only one cotyledon, which is quite thin and specialized, called a scutellum. Its main job is absorbing nutrients from the endosperm and passing them to the embryo. Maize seeds also have these clever protective sheaths, a coleoptile covering the young shoot and a coleorhiza covering the young root.
What do those do?
They protect the delicate growing tips as they push up through the soil after germination. Very important for monocots.
Now, once a seed is formed, it often doesn't sprout right away. It goes into this state of dormancy. It's like the plant hits a pause button, waiting for the right moment.
Exactly. And the cues to break this dormancy are incredibly varied and wonderfully adapted to the plant's environment. Seeds from desert plants might only germinate after a really substantial rainfall washes away inhibitory chemicals.
Makes sense.
Others need intense heat, maybe from a wildfire passing overhead, which cracks a seed coat. Some need smoke chemicals. Seeds from plants in colder climates often require stratification, a prolonged period of cold, to ensure they don't sprout in autumn only to be killed by winter.
Wow.
Some even need to pass through an animal's digestive tract. The acids weaken the seed coat. It's amazing. There's that famous example of a nearly 2,000-year-old date palm seed found at Masada in Israel that was successfully germinated. Dormancy ensures seedlings emerge when conditions are most favorable for survival.
That's a truly ancient pause button. Okay, so once that button is released, germination begins. How does that kick off?
It usually starts with imbibition, the seed taking up water. This causes the seed to swell, which often ruptures the seed coat, and it rehydrates the dormant embryo, triggering metabolic changes. Growth resumes.
And what comes out first?
Typically, the very first thing to emerge is the radicle, the embryonic root. It grows downwards, anchoring the seedling quickly and starting to absorb water and minerals from the soil.
Smart move. Get the water supply sorted first.
Absolutely. Then the shoot needs to break through the soil surface. And amazingly, different plants have evolved distinct strategies for this delicate maneuver. Many eudicots, like your garden bean, form a protective hook in the hypocotyl, the part of the stem below the cotyledons. This hook pulls the delicate cotyledons and the embryonic leaves safely up through the soil.
Like pulling them up backwards.
Sort of, yeah.
Right.
It protects the fragile shoot tip. In contrast, monocots like corn use that protective coleoptile sheath we mentioned. It pushes straight up through the soil like a little spear, and the true shoot tip then grows up through the tunnel it creates.
Very different strategies. Okay, finally, the third F, fruits. We've established that a fruit is essentially a mature ovary, protecting the seeds and helping disperse them. Fertilization triggers hormonal changes that cause the ovary wall to thicken and mature into the pericarp.
Right. The pericarp is the technical term for the fruit wall, which can be fleshy or dry. And what's truly fascinating is the sheer diversity of fruits, often classified by how they develop. Simple fruits, like a pea pod or a cherry, develop from a single carpel or several fused carpels of one flower.
Okay.
Then there are aggregate fruits, like a raspberry or a blackberry. These develop from a single flower that had many separate unfused carpels. Each tiny little sphere on a raspberry is technically a fruitlet derived from one carpel, all clustered together.
Ah, that makes sense why they look like that.
Exactly. Multiple fruits are even more complex. Think of a pineapple. That develops from an entire inflorescence, a group of tightly clustered flowers, whose ovaries all fuse together as they mature into one large fruit. And then you have accessory fruits. In these, other floral parts besides the ovary contribute significantly to the fleshy part we eat. An apple is a classic example. The core is the ovary, but the fleshy part we eat is derived mainly from the swollen base of the flower. Strawberries are similar. The fleshy part is the receptacle, and those tiny seeds on the surface are actually individual fruits called achenes, each containing a single seed.
That is wild. So an apple isn't just the ovary.
Nope. Mostly floral base. It's a testament to evolutionary tinkering and ingenuity.
And the ripening process itself is a masterclass in biological engineering, especially for fleshy fruits. It seems designed to attract animals.
Absolutely. It's a symphony of hormonal changes. Enzymes break down cell walls, making the fruit softer. Chlorophyll breaks down, revealing yellow or red pigments. Starches and acids are converted into sugars, making it sweeter. All these changes, color, texture, taste, aroma, are signals saying, eat me, to potential animal dispersers.
Turning them into unwitting but highly effective seed delivery services.
Indeed. And these dispersal mechanisms are incredibly varied. We've talked about wind-think dandelions with their parachutes, or maples with their helicopter wings. Water dispersal works for things like coconuts, which are buoyant, but animals are probably the most common dispersers.
Like the burrs we started with.
Exactly, clinging onto fur, or animals eating tasty fruits and then defecating the seeds elsewhere, often with a little fertilizer bonus, or squirrels and jays hoarding nuts and acorns and forgetting where they buried some. Some plants even produce little food bodies called elaiosomes on their seeds specifically to attract ants, which carry the seeds away. Every mechanism is a strategy for the next generation to find a new place to grow.
So plants are clearly masters of sexual reproduction, with all those intricate steps. But you mentioned earlier that some plants have found a way around all that fuss, essentially making copies of themselves. How does that work?
Exactly. And that's the equally remarkable world of asexual reproduction, often called vegetative reproduction in plants. This process produces offspring that are genetically identical clones of the single-parent plant without any fusion of egg and sperm.
Like making cuttings.
That's one way humans exploit it, yes. But plants do it naturally too. Think of fragmentation. A piece breaks off the parent plant and develops into a whole new individual. Like a piece of potato with an eye that's a bud can grow a whole new plant. Or those tiny plantlets that form along the edges of a kalanchoe leaf, they drop off and root.
Right, I've seen those. And you mentioned aspen groves.
Yes, some aspen groves are absolutely enormous, covering many acres, but genetically they are a single individual. Thousands of trees, all connected by one massive underground root system, periodically sending up new stems. It's mind-boggling. And then there's apomixis, which is really fascinating, found in plants like dandelions. They can produce seeds, but the embryo develops without fertilization directly from a diploid cell in the ovule. So they get the dispersal advantage of seeds, but the offspring are still genetic clones of the parent.
Kind of cheating the system.
In a way, it combines dispersal with cloning.
So what are the big trade-offs here? Why would a plant choose asexual versus sexual reproduction? Or maybe do both?
That's an excellent question because each strategy has clear pluses and minuses. Asexual reproduction is great if the plant is really well suited to its current stable environment. Why change a winning formula right now?
Sure.
It also doesn't require a pollinator, which is a huge advantage if plants are sparsely distributed or pollinators are scarce. Plus, offspring produced from mature vegetative fragments, like a piece of stem, are often stronger and more established than tiny seedlings initially.
Okay, sounds good. What's the downside?
The major disadvantage is the lack of genetic diversity. Because they're all clones, if the environment changes, say a new disease pops up or there's a drought, the entire population could be wiped out because they all have the same susceptibility. There's no variation for natural selection to act upon.
So if asexual reproduction leads to vulnerability, then sexual reproduction must be the plant's strategy for adaptability.
Precisely. Sexual reproduction's biggest advantage is generating genetic variation in offspring through meiosis and fertilization. This diversity is absolutely crucial for evolutionary adaptation, especially in unstable or changing environments, or when dealing with co-evolving pathogens and pests.
Rolling the genetic dice.
Exactly. It increases the chance that at least some offspring will have traits that allow them to survive and reproduce under new conditions. Also, the seeds produced sexually often facilitate dispersal over longer distances, and they can remain dormant, waiting for favorable conditions, which vegetative fragments usually can't do.
Okay, so diversity is key. Given those benefits, how do plants avoid fertilizing themselves, especially if a flower has both male and female parts? How do they ensure genetic mixing actually happens?
Great question. Plants have evolved numerous ingenious mechanisms to prevent or reduce selfing. Some species are dioecious, meaning individual plants are either male, producing only pollen, or female, producing only ovules. Think of holly trees. You need both male and female trees to get berries.
Oh, okay.
Other plants, even with perfect flowers containing both stamens and carpels, might have mechanisms like maturing their pollen at a different time than their stigma becomes receptive. Or the physical arrangement of the parts makes self-pollination difficult, like the stigma being positioned far above the anthers.
Clever structural tricks.
But the most common and genetically sophisticated mechanism is called self-incompatibility. This is a biochemical system where the plant recognizes and rejects its own pollen, or pollen from very closely related individuals. It's based on alleles of specific genes called S genes. If the pollen grain shares an S allele with the pistil it lands on, fertilization is blocked.
Like a lock and key system that only works with different keys.
Kind of, yeah. It's like an immune system for reproduction, ensuring outcrossing and genetic mixing. There are different types, gametophytic and sporophytic, based on whether the pollen's S allele or the parent plant's S alleles control the rejection, but the result is the same, promoting diversity.
And this inherent flexibility in plants leads us to something truly mind-boggling, to typidency. You mentioned this briefly, the ability of a single plant cell, theoretically, to divide and develop into an entire clone of the original organism.
It really is a remarkable ability, not common in the animal kingdom to that extent. And humans have been leveraging this totipotency for centuries, even millennia, to propagate plants vegetatively. Taking cuttings as a prime example. The cut end forms a mass of undifferentiated cells called a callus.
Right.
And then adventitious roots that arise from non-root tissue, like the stem develop from it, creating a new genetically identical plant. Grafting is another ancient technique. Joining a scion, a twig or bud from a desirable plant onto the stalk, the root system of another plant, often chosen for resilience or soil adaptation.
Like with fruit trees or grapevines.
Exactly. It allows us to combine the best traits of two different plants into one functional unit.
And modern science takes us even further with test tube cloning, right?
Yes, plant tissue culture. You can take tiny pieces of plant tissue, sometimes even single cells, and culture them on a sterile nutrient medium in a lab. Under the right conditions, they form that undifferentiated callus. Then, by carefully manipulating plant hormones in the medium, you can induce that callus to develop roots and shoots, eventually growing into a complete plantlet, a clone of the original.
What's the main advantage of doing it that way?
Well, it allows for rapid propagation of large numbers of plants from a small amount of starting material. Crucially, it can also be used to produce virus-free plants. Often, the apical meristem, the very tip of the growing shoot, is free of viruses even if the rest of the plant is infected. By culturing just that meristem tip, you can regenerate healthy plants.
That's clever.
And perhaps most importantly for our next topic, test tube cloning is absolutely essential for genetic engineering in plants. You can introduce new DNA into plant cells in culture, select the cells that have successfully incorporated the new gene, and then use tissue culture techniques to regenerate a whole genetically modified plant from that single modified cell.
It's clear that humanity has played a huge role in modifying crops, long before modern labs. Think about corn or maize. You said modern maize literally cannot disperse its own seeds.
That's right. The kernels are tightly packed on the cob and enclosed in husks. It relies entirely on humans to separate and plant the seeds. It's a product of thousands of years of artificial selection by early farmers in the Americas, starting perhaps 10,000 years ago, from a wild grass called teosinte that looks drastically different.
So we've been genetic engineers for millennia, in a way.
In a practical sense, yes, selecting for desirable traits generation after generation. And it's important to remember that natural hybridization, like the complex crosses between different wild grass species that eventually led to modern wheat, also provided a rich genetic foundation that early breeders worked with, even if they didn't understand the genetics.
And traditional plant breeding, as it developed into a more formal science, involves carefully selecting parent plants with desirable traits, say, high yield and disease resistance, and then hybridizing them, crossing them.
Right, but it often takes many generations. If you cross a high-yield crop with a wild relative to get a disease resistance gene, you also bring along lots of undesirable traits from the wild plant. You then have to do repeated backcrossing to the elite crop variety to gradually get rid of the unwanted genes while keeping the one you want. It's slow, painstaking work.
This is where modern genetic engineering or biotechnology really offers a different approach.
Exactly. Genetic engineering allows for the direct transfer of specific genes, often just a single gene, sometimes even between completely unrelated species, something impossible with traditional breeding. You could potentially take a gene for cold tolerance from a fudge and put it into a tomato plant, for example.
Wow.
It's much faster, much more targeted, and it bypasses the need for intermediate crosses or sexual compatibility. Organisms modified this way, containing genes from other species, are called transgenic.
And the promise of these genetically modified or GM crops is huge, isn't it?
Yeah.
Especially when we think about global challenges like food security and climate change.
The potential is certainly significant. We're already seeing benefits in reducing hunger and improving agriculture. Take bat maize, or bat cotton. They contain a gene from the bacterium Bacillus thuringiensis that produces a protein toxic to certain insect pests.
So they protect themselves from bugs.
Precisely. This reduces the need for chemical insecticides, which is better for the environment and farmer safety. And interestingly, bat maize often has much lower levels of fumonisin's nasty toxins produced by fungi that often infect insect-damaged corn, making it potentially safer for consumption.
That's an unexpected benefit.
There are also herbicide-tolerant crops like Roundup-ready soybeans or corn. These allow farmers to spray herbicide over the whole field, killing weeds without harming the crop. This facilitates no-till farming, which significantly reduces soil erosion. We also have disease-resistant crops, like the transgenic papaya engineered to resist ring spot virus, which literally saved Hawaii's papaya industry.
Incredible. And what about improving nutrition?
That's a major area too. Golden rice is probably the most famous example. It's engineered to produce beta-carotene, a precursor to vitamin A in the edible grain. Vitamin A deficiency is a huge problem in many parts of the world, causing blindness and death, especially in children. There's also work on biofortified cassava, engineered for higher iron and beta-carotene, and reduced levels of natural cyanide-producing compounds.
Beyond food, you mentioned GM crops could help with our reliance on fossil fuels.
Yes, biofuels are another big focus. Scientists are engineering fast-growing plants like switchgrass and poplar trees to have modified cell walls. Specifically, they're trying to reduce the amount of lignin, a complex polymer that makes cell walls rigid but also very difficult to break down.
Making them easier to convert to fuel.
Exactly. Reducing lignin makes it much easier and more efficient to convert the plant's cellulose into sugars, which can then be fermented into ethanol. The goal is to create efficient, sustainable, and ideally carbon-neutral sources of liquid fuel from plant biomass.
Despite all these incredible prospects, there's obviously a significant ongoing debate and public concern surrounding genetically modified organisms, GMOs, particularly the idea of releasing them into the environment.
There are indeed legitimate biological concerns that need careful consideration and rigorous scientific assessment. One area is potential risks to human health. Some people worry about introducing new allergens. Biotechnologists are aware of this and screen for and try to remove genes encoding known allergenic proteins.
Right.
And as we mentioned, with butt maize having less fumonisin, sometimes GM foods might actually be healthier in certain aspects. The debate over mandatory labeling of GM foods reflect these complex discussions about consumer choice and perceived risks.
What about the effects on other organisms, the non-target effects? That was a big concern with early studies on monarch butterflies and butt maize pollen, wasn't it?
That's right, and it was an important case study. An initial lab study suggested that monarch butterfly larvae could be harmed by eating milkweed leaves dusted with butt maize pollen. It caused quite a stir.
I remember that.
However, subsequent, more extensive field studies painted a much more nuanced picture. They found that the amount of pollen naturally landing on milkweed in and around cornfields was generally below levels that caused harm. They also clarified that the initial lab study might have used higher concentrations or other plant parts mixed with pollen. Importantly, large-scale studies suggest that the impact of insecticide spraying on non-blight corn acreage is actually a much greater threat to monarch populations than blight corn itself. It really underscores the need for realistic, field-based risk assessments.
Okay, so context matters hugely, and then there's the concern of transgene escape, the idea that genes introduced into a crop might spread to wild relatives through cross-pollination, potentially creating herbicide-resistant superweeds, for instance.
That is a valid ecological concern, especially in areas where crops have compatible wild relatives growing nearby. If an herbicide resistance gene got into a weedy relative, it could make that weed much harder to control.
What strategies are being explored to prevent that from happening? Are there biological ways to contain the genes?
Yes, scientists are actively working on several containment strategies. One approach is engineering male sterility into the transgenic crop, so it produces viable seeds if pollinated by something else, but its own pollen doesn't carry the transgene or isn't functional.
So it can't spread the gene via pollen?
Correct. Another idea is to transfer apomixis, that asexual seed production we talked about, into transgenic crops. They could then be propagated by seed, but the seeds would be clones and wouldn't involve pollen transfer. A third strategy is engineering the transgenes into the chloroplast DNA instead of the nuclear DNA. In many plant species, chloroplasts are inherited only maternally, through the egg cell, not through pollen.
Ah, so the pollen wouldn't carry the gene.
Exactly. And a fourth, perhaps more complex approach is engineering flowers that simply fail to open, or that are modified to ensure only self-pollination occurs, physically preventing pollen from escaping into the environment.
It sounds like there are multiple potential solutions being researched.
Definitely. It's clear that decisions about using biotechnology, particularly releasing GMOs, involve carefully weighing the potential benefits against the potential risks. It's a complex equation that really demands ongoing research, rigorous testing, case-by-case assessment, and decisions based on sound scientific information rather than just blanket acceptance or rejection.
Wow, what a deep dive into the plant kingdom. We've journeyed from a dog's fur in the Alps all the way to the cutting edge of genetic engineering, all thanks to the humble plant. We've uncovered the incredible adaptations of angiosperms, their intricate life cycle involving the three Fs, flowers, double fertilization, and fruits, and the profound ways humans have interacted with and modified them over millennia.
We truly have. From the elegant mechanics of their reproduction, encompassing those diverse sexual and asexual strategies, to the transformative power of both traditional plant breeding and modern genetic engineering. Well, it's clear that plants are not just static green things, they are dynamic biological systems. They've shaped our world and continue to offer potential solutions to some of our biggest challenges, while also presenting new questions and ethical considerations to grapple with.
So next time you bite into an apple, or maybe even just walk past a dandelion pushing through the pavement, take a moment to appreciate the hidden biological wonders at play. There's a whole universe of complexity, adaptation, and, frankly, genius in every leaf and seed, constantly evolving, constantly surprising us.
And maybe a final thought to leave you with. As we continue to unlock and increasingly manipulate the genetic blueprint of plants with ever greater precision, how might our capabilities challenge our very definitions of what we consider natural? And how might this reshape the future of agriculture, ecosystems, and maybe even our relationship with the living world in ways we can barely begin to imagine today?